Karen for the introduction. I am very happy to present our recent achievements in CRISPR Cas9 modified inducible pluripotent stem cell for in vitro disease modeling. This topic becomes very hot due to the rise in genome editing technology and the progress in stem cell technology. So today, my topic will cover current trend in gene-modified iPS cells as genetic disease models. Our technology platform for iPS cell gene modification. Applied stem cell has been in this field for more than 7 years. We have accumulated a lot of experience, and I would like to share with you our successful cases. We also have collected several iPS cell disease model panels, which I will also share with you today. Finally, I would like to give you an example of using an iPS cell disease model for potential disease treatment. So hopefully you will get satisfied by all these topics today. So most of you probably already saw a similar figure like I show here. The idea is that we obtain the biosamples such as skin or blood from patients with injury or disease. The samples, for example, fibroblasts, can be reprogrammed into iPS cells. At this stage, the iPS cells can be genetically modified and further differentiated into different lineage cells, such as neurons, cardiomyocytes, beta islet cells, and hepatocytes. These tissue-specific cells with corrected mutation can be used for autologous transplant, or the lineage cells differentiated from paired isogenic iPS cell lines can be used for disease research or drug screening. So what is the advantage of using iPS cells? If compared with primary cells, the iPS cells provide unlimited source of human tissues carrying genetic variations. In addition, patient's source can be limited. Isogenic lines from healthy donors can mimic patient-derived iPS cells by introducing mutations. Another advantage is that we can use isogenic iPS cells from patient to avoid genetic variants. One more important consideration is that iPS cells are relatively easy to handle and to be genetically modified if compared to primary cells. We all know that primary cells have short lifespan and incapability of single-cell cloning. iPS cells allow edited gene exist in lineage cells. So to establish the iPS cells disease models, applied stem cells uses the proprietary stem cell technologies, which enable us to generate iPS cells from healthy individuals or patient samples, starting from skin biopsy, BPMC, and other biosamples, and then differentiate them into lineage-specific tissue cells with high purity. Using our genome editing technologies, we can generate genetically modified cell lines, especially focusing on stem cells. Others also, we can include the cancer cell lines, customer preferred cell lines, and animal models, for example, mice and rats. In this slide, I summarized our technology platform. We used nuclear reprogramming factors licensed from iPS Academia Japan for iPS cells generation. And then when iPS cell generated, we used the CRISPR Cas9 technology licensed from Broad Institute and the target from Stanford University to modify the iPS cell genetically. And the iPS cells, either gene modified or the original iPS cells, can be further differentiated into lineage-specific cells. And at this stage, we can use our own IP, which is iPS cell differentiate into lineage-specific cells with high purity. So all this well-patent protected platform can fully protect your investigation outcomes. Here, I would like to give you some background of our two genome editing technologies. Target on the left side is an integrase-based genome editing tool, which we licensed from Stanford University. This technology requires pre-engineering a docking sites we call ATTP at safe loci in the genome. The targeting vector contains the gene of interest and a recognition site we call ATTB, which can recognize the docking site ATTP. With the enzyme, here we use 5C31 integrase. Gene of interest will be integrated into the safe harbor. Target is a very good tool for inserting large fragment DNA. On the right side is a summary for CRISPR Cas9 technology that we get the rights from MIT. So CRISPR Cas9 is a very good technology for making gene knockout, point mutation, and small DNA insertion. The Cas9 directed by guide RNA can generate double-strand DNA break and introduce an indel. With or without donor DNA, CRISPR-Cas9 can make knock-in or knock-out mutation at the specific site directed by guide RNA. By comparing the two technologies, we see that there are two complementary technologies, with CRISPR very efficient for knockout, point mutation, and small fragment knocking. At the same time, Target is more efficient for large fragment knocking. Translating into gene editing in iPS cells, what are the current difficulties, even with our advanced gene editing technologies? We know that patient iPS cells are highly variable and prone to differentiate. Different culture systems, I will give you an example later, can also generate variability in targeting efficiency. Large-scale cell line evaluation, screening, and genotyping, as well as difficulties in single-cell cloning. All these require tedious work, time and labor-consuming. So for fix all this problem, what is our solution? Here I would like to give you an example how we increase the targeting efficiency with cell line pre-evaluation. We first optimized the culture systems. So we used math-based systems and the feeder-free systems, and we tested different transfection conditions. So first, we would like to show you the data between the two culturing systems. On the left, there are two transfection conditions using math systems. If we compare to the same transfection conditions with feeder-free systems, we saw that the efficiency for transfection is dramatically improved in the feeder-free systems. So the conclusion is feeder-free systems is better for transfection efficiency on our hand. So we determined that feeder-free system is better compared to math systems. Then we compared 5 different conditions, transfection conditions, in the feeder-free systems. And the 5 conditions, among these 5 conditions, the condition 2, 4, 5 were identified as better transfection conditions for further evaluation. We then compare these three transfection conditions with cell death. From the figure showing here, the condition 5 had less cell death and therefore was selected for gene targeting. So this slide actually we are trying to testing the drug killing concentrations. So we have selected 5 different concentrations named 0.25x, 5.5x, 1x, 2x, and 4x, and compare them to the control. So the picture shows here clearly tell us that 1x concentration is the best drug concentration that we can use for further study. So here I would like to give you a summarization for the transfection and culturing condition optimization. So we look at the transfection efficiency before and after optimization at different conditions, and also the overall targeting efficiency. So from the data I showed you earlier, with the improved culturing condition, the transfection efficiency can be improved by 70%. The transfection condition optimization improves the efficiency by 30%. This improvement lead to an overall increase in the targeting efficiency by 50%, especially in the case of large fragment knock-in using CRISPR. So in the following couple slides, I will walk you through 2 examples that we use CRISPR to make knockout and point mutation in iPS cells. Here is a case of gene knockout in normal iPS cells to mimic patient-derived iPS cells carrying a pathologic disease, mutation. When we are working on cell line evaluation, we usually start the guide RNA design and construction and validation at the same time. So we chose at least 2 guide RNAs with the best off-target profile to test in human cell lines. This is to ensure that the guide RNA works for human genome. The in vitro testing results are shown here. Between the 2 guide RNA, G1 and G2, obviously that G2 had higher efficiency and therefore was selected for future study. Potential positive clones were genotyped, first by enzyme digestion. Here is the gel picture. We selected 10 positive clones we selected 10 clones for the here, and we saw that 2 seems like heterozygous and 3 are homozygous. So the efficiency is about 50%. And the positive ones were sent out for sequencing to confirm that the designed mutation was correct. So the designed mutation then confirmed by the sequencing. I didn't show the data for the sequencing. So just to let you know that we will finally sequence this positive clones to make sure the mutation is right. So there is another example I list here is a point mutation correction in patient-derived iPS cells. Similarly, the guide RNA was designed based on the mutation site to be corrected. The guide RNA would then functional validate in a human cell line. For this particular case, guide RNA number 1 was selected, which had a better cutting efficiency. For making point mutation, we need a single-strand donor DNA, and in this case, it was designed to introduce the point mutation to the specific site, which is indicated in red. So for a point mutation correction, we need to using sequencing, PCR and sequencing to verify the results. So the positive clones were sent for sequencing. By comparing the patient's sequence on the top and the corrected sequence at the bottom, we saw that the point mutation was corrected as we read. So these are the two examples that we use CRISPR for iPSC gene modification. As I mentioned earlier, besides CRISPR, we also have another technology target is also important to perform gene editing. So just to give you a reminder in these slides, how this technology works. Again, the system requires a docking site in the genome, a recognition site in the targeting vector, and the technology works for large fragment knock-in with high efficiency. The idea of using target in iPS cells is first to generate a masked iPS cell line. What is a masked iPS cell line? So to make this iPS cell master cell line, the ATTP site will be inserted into a safe harbor, such as H11 or ROSA 26. The ATTP recognition sites and the gene of interest, for example, GFP here, will be constructed in the targeting vector. The cells with successfully insertion express GFP and turn in green. By using the master iPS cells, we have been successfully inserted an M-cherry report gene. The PCR gel picture indicates that 12 out of 23 clones are positive with the right insertion. The efficiency is more than 50% without drug selection. So this is a very impressive results for using target. Another application is to use the masked cell line and direct differentiate them into lineage cells. Here, we used a nesting promoter, a neuron-specific promoter, to drive expression of 4 membrane-specific transcription factors, plus a report gene M-cherry. We started from patient-derived and normal masked iPS cell lines with ATTP at H11 locus. The successful insertion drove the iPS cells into dopaminergic neurons, and it turns into red. We currently have the masked iPS cell lines derived from a normal individual with ATTP at ROSA 26. So for using this masked line, we can make report iPS cells lines, differentiate the iPS cells into lineage-specific cells, expressing lineage-specific markers or reporters for cell enrichment and purification. Here is a table that I summarize the targeting efficiency of gene editing in iPS cells using different technology. So I listed the major two technology, CRISPR and target. Like I said before, CRISPR is perfect for knockout, for mutation, and small fragment knock-in. So using CRISPR for generating these mutations without drug selection, the average efficiency is about 30% and 10% respectively. So for large fragment knock-in, we can use either CRISPR or target, but target will have more capability for inserting even larger fragment. So with drug selection, the CRISPR Cas9 can generate almost a 60% target efficiency, while target can have 100% target efficiency with drug selection. So with all these technologies and all this experience, what we can offer you for the existing disease models? Like I said, applied stem cell has been in this field, stem cell gene editing field, for more than 7 years. We have collected several disease panels using iPS cells. We have ALS8 carrying VAPB gene mutation from 2 families. One contains 2 controls and 2 patients, and the other contains 1 control and 2 patients. We have 2 mutation-collected isogenic lines. We also have isogenic lines from Parkinson's patients and other neurological disease. So if you have any interest, please inquiry to our website or using our email address and phone number. For using iPS cells as disease model, one critical consideration is the phenotype readout. Here, I would like to give you an example how scientists using iPS cells disease models to derive iPS cells and the mutation-corrected lines were used. So in ALS disease, we know that neurofilament aggregation is a hallmark pathology. So this NF aggregation was used for this study. We saw that on the left panel is in the motor neurons that differentiate from the patient iPS cells. There is clear shows NF aggregation indicating by arrows and arrow hands. However, on the right side is the motor neurons corrected from the patient iPS cells, which carries normal genotype. So we saw that the NF aggregation is significantly decreased in the corrected ALS iPS cells. So for conclusion, this data suggests that using lineage-specific cells differentiated from iPS cells could be a useful model for developing new treatment. As a summary, applied stem cell can provide the whole platform for gene editing in iPS cells and then differentiate them into lineage-specific cells. So we can provide high-quality iPS cell disease modeling services, like we can starting from generating the iPS cells using footprint-free method with high efficiency. We modify the genes in iPS cells using advanced technologies, CRISPR and Target. We differentiate iPS cells into tissue-specific cells with high purity. We offer iPS cells disease model panels for drug screening. Okay, I think that's all for today's topic. Now I would like to take some questions from you.
Thank you, Lu Ping, for the informative presentation. And thank you all for sending us your questions. We will try to answer them here now. Okay, the first question. What kind of methods do you use to generate iPS cells?
Okay, we have different options. So we have footprint-free method like episomal mRNA. And for other researchers that who like to follow Yamanaka's original paper, we can also use retrovirus system to generate iPS cells. So we have different options for all your needs.
Okay, thank you. What's the advantage of Target over CRISPR to make gene knock-in cell lines?
Okay, that's a very important question. So I've been focusing on this topic in the whole presentation, and I would like to take the chance to give you a summary. Target is very efficient for knocking, especially large fragment knocking. So we have successfully inserted a 22 kb fragment without drug selection. And we are still trying to insert even larger fragment using this technology. And with a selective marker, as I mentioned, efficiency could be 100%, and without a selective marker, the efficiency could be 50%. So this is the first advantage of Target over CRISPR, is the high efficiency for large fragment gain. And the other advantage is, like I mentioned, we can either use selective marker or not using selective marker. So in either way, the construction design and the steps involved for construction, vector construction, is simpler compared to the CRISPR. And that's why we need less optimization steps and save cost and time. So there are the two advantages: high efficiency and cost and time saving compared with CRISPR.
Okay, the next question is what QC steps are involved after gene modification in iPS cells?
QC steps is very important, so I like this question. There usually have two type of QC steps, which one is generally for all gene editing using CRISPR. That is the off-target profile. So we check in the off-target profile as a step, as a standard procedure for all iPS cell gene editing and other cell line or animal gene editing. And particularly for iPS cells, we have another two options, which are the karyotyping and the pluripotency characterization. So for karyotyping, we can perform the G banding or chromosome counting. And for pluripotency characterization, we can do marker staining, EB formation, and teratoma analysis to ensure that the final deliverables of iPS cells after gene modification is qualified for your further study. And I think this is very important for you.
Okay, the next question, what is the success rate of inserting a 6 kb fragment using CRISPR?
6 kb is pretty feasible using CRISPR with a drug selective marker on our hands. So the efficiency is about 60%.
Okay, and looks like there's one more question here. How many times can cells be passaged after iPS gene modification?
Okay, this is a little complicated question because the answer really depends on the initial status of the iPS cells. So if we use patient-derived iPS cells, normally the patient-derived iPS cells grow relatively slow and are prone to differentiate. So it's hard to predict. However, we do actually have the pre-evaluation and optimization steps, which we think is very critical. So in this way, we can understand the cell line better, and we can handle and maintain them better to make sure that the iPS cells, even after gene modification, can culture longer. And to give you an idea, if we use our normal iPS cells to perform gene editing, we usually can go further to 60 passages for even after gene modification. So this number just gives you an idea if we're using our normal iPS cells.
Okay, thank you, Luping, and thank you all. That's about all the time we have for questions at this moment. For additional questions that we didn't get to answer here today, we would be happy to answer every single question that you send us by email. Please just go ahead and email those to info@appliedstemcell.com, and we'll answer them for you. And we will also send you a copy of the presentation along with a short survey. And please let us know if there are any other webinar topics you would like to hear about in future presentations. We surely appreciate your feedback. And also, I just also want to let you know we have a year-end discount promotion going on right now on our genome editing services discussed here. And that goes till the end of the month, end of the year here. And please call or email us with your inquiry for those at the above same email address shown here. Thank you again for attending Applied Stem Cells iPS Genome Editing webinar. And we look forward to hearing from you soon.